receptor page. So that you can find on the receptors, the receptor page here. And at first, for most tools, you're presented with a selection option. So you have to, in some way, select what receptor you're interested in. Where do you want to know more about? We have the browse option, where you can go through each of the classes of the GPCR, and then, for example, via the ligand types, and then each of the subfamilies, you can go down to each individual receptor and select the receptor of interest. Or you can directly search, for example, for a specific receptor if you know what you're exactly looking for. What's also good to note, and something that not all people are aware of, is that we do have these filter options here at the top. So by default, we're selecting and filtering on humans, but actually we have a lot of different species in the GPCRdb. So you can actually also select all species, or if you want to specifically select one, we have a whole long list of different species that you can go through and select the one of interest. Another important one is the source. So, for example, most human, all human GPCRs, they have been curated and annotated, and that makes them part of the SwissProt part of the UniProt database. So that means that they're completely reviewed and curated. But several of the sequences, they are not automatically embedded. So they're not yet reviewed. So that's what you can toggle here. So you can also show unreviewed GPCR sequences by going from SwissProt to all sources. Okay, so for this one, I'm just gonna go to an example. Let's take one of the prototypical GPCRs, the beta-2 adrenoceptor. And this is the most well-known page, I would say, of the GPCRdb. So this is the receptor information page. Just to note, I've now gone through the menu system and the whole selection process. Here at the top right, we also have a very quick search option. So if you don't want to go through all these options and you know exactly what you're looking for, you can also type it in here. For example, you're looking for the adrenoceptors, or you're looking for the beta-2, and then you can find them here. Or if you already know exactly what UniProt entry name you're looking for, then you can type it in here. By default, also here, the human entries are returned, but if you know the exact entry with the species, then you can type it in here, and then it's also automatically selected. So then back to the information page. At the top, you can find a small summary of this receptor. So you can find, for example, that the beta-2 adrenoceptor is part of the class A, the rhodopsin class. It falls under the aminergic receptors, and it's part of the subfamily adrenoceptors. And it has different gene symbols, the official ones and some previous ones that have been used. More information about the human, about the organism, and alternative naming and the source where it's coming from. So that's Tremble or SwissProt. Then here we have the sequence overview. The sequence overview provides already quite a lot of information. So instead of just having a long list of amino acid codes, it shows you in green and in blue what elements are actually the termini and the loops, and what are the DM helices. Moreover, if you're hovering over the receptors, the residues, then you can see more information about that specific residue. You can see that it's, for example, in transmembrane helix 1, it's residue glutamate 30, and it's at position 129 according to the Ballesteros-Feinstein numbering. And according to the structural numbering of the GPCRdb, so that's highlighted after the X, it's also at position 29. So that's the usual numbering. But the advantage of the GPCRdb numbering is highlighted when, for example, we know from structures that there are bulges and constrictions in the helix, because that actually means that although normally, according to the Bioceres-Weinstein numbering, a residue should be at a specific position, that right now is that is actually at a different position. So, for example, in the case of beta-2 adrenergic, the serine at 546, that forms a bulge. So it's actually between the residue displacement of 546 and 547. So that's why we have this adjusted numbering scheme in which there is a correction for the structural characteristics of the receptors. An extra feature on top of this is that we did not only annotate the transmembrane helices, but also for other several conserved parts in the GPCRdb, in the GPCR structures, we've also created an additional numbering scheme. So, for example, in intracellular loop 2, we have this numbering scheme 3, 4, and then dot, and then this conservation numbering again. So that actually enables you to compare also the loop regions between different receptors. So that's just the sequence part. And then below you can find direct links, for example, to Campbell to find more information about the beta-2 adrenoceptor, the guide to pharmacology, and also UniProt. And below that we have the famous snake plot and the helix box plots. To use this, we have predefined properties, predefined sets of colorings. So, for example, if you just want to show what type of residues are at which position, you can color them according to properties. So, for example, the hydrophobic residues, and then the aromatic ones, and the more polar ones. But we also have the option to show which in vitro mutations are already known for these receptors. And if you then hover across one of these receptors, you also get a nice summary. So, for example, we know that for exactly this position, we have data for 3 mutations, 3 unique mutations, so more data points, and several of them actually resulted in a decreased affinity of the ligand, some and selected other actually resulted in no change. So that already provides you with quite a lot of information. With these buttons, we also have information about post-translational modification sites, and then also you can hover here about over the residue again and find what type of modification has been annotated for these residues. And the same goes for natural genetic variations that have been observed. So, for example, what type of mutations have been observed and where, and what effect this was predicted to have on the receptor. And the last one is to show interactions that have been observed from structures. So if we have structures for this receptor, then we collect the interactions that ligands have with the receptor, and then it's highlighted here which residues actually interact with these ligands. And if you hover across it, you can directly see again how many times an interaction was observed and also what type of interaction. And this is not the only thing. It could, for example, also be very interesting for your publication. If you're making a publication about specific segments, that you use this and that you, for example, color some of these residues yourself, and that you then also highlight which ones were the most important ones, and what was crucial, for example. So you can easily create a figure for your publication and then download it in different formats depending on what you like to work with, including the SVG format, so you can also change it, for example, using Photoshop. For the snake plot, it actually works in the same way. So we have the same options here. So we have you can color the option, the residues in the same way. You have the same coloring options here, and the last one is actually one that you can use to find also custom colors and then color it according to your most your favorite color. What most people might not automatically see about the snake plot is that actually these loops and the termini are interactive. So if you click on it, actually they are drawn automatically. So depending on what you want to highlight and what you want to remove from the snake plot, you can dynamically change your snake plot. And then at the end, you can download it again. At the bottom, we have the mutation data again, and information about the endogenous ligands, homology models that we have created for this receptor. It's good to know that we only create homology models for the receptor and states that were not yet crystallized or for which we don't have experimental data. And for the experimental structures, you can directly go to structures here.